Hello, everyone, and welcome to phyloseminar.org. The current theme is tree sequences, and this is the third talk in a series of 3 talks on that topic. Please use the YouTube live comment box to ask questions or tweet at phyloseminar. Today's speaker is Leo Speidel. Leo is a postdoc in Simon Meyer's group at the Department of Statistics, University of Oxford, where he completed his PhD last year. His PhD focused on developing RELATE, a method for inferring genealogies genome-wide for many thousands of samples. Currently, he is working on extending this framework as well as developing powerful methods that take inferred genealogies as input to answer a broad range of questions relating to population genetics. Thank you, Leo, and welcome to phyloseminar.
Yeah, thank you very much. So thank you for having me here today, and thank you for tuning in. So this is part 3 of 3. And so the focus of this talk will be about what can we learn from inferred genealogies and tree sequences. We've worked quite hard in part 1 and 2 to sort of get these genealogies, and now we want to see like what kind of information can we read off from these genealogies. Okay, so perhaps we can start with a fairly sort of obvious or basic point, namely that genetic variation in a population of individuals is shaped by quite a long list of complex sort of evolutionary processes. And so some of these are more on the molecular level. Mutation obviously first gives rise to genetic variation, and then recombination sort of shuffles this up a little bit in every generation. So these are sort of the more molecular types of processes, but then there are other processes that are sort of more on the macro scale. So for humans, we can think of sort of human migrations, population bottlenecks, and mixture events, and so on. And I think it's also quite nice to always remember that all of these processes might themselves evolve. So for instance, we can think of selection pressures changing depending on the environment and so on, and mutation rates evolving through time, and yeah, similar things for essentially every type of process that shapes genetic variation. Okay, and so our goal is to make inferences about these processes or events in our past, given data of genetic variation that we typically collect either at the present day or sort of sometimes at sort of time points in the past, but typically, so we'll have data here at the tips of these genealogical trees, and these tips, as these genealogies, will relate these individuals back in time, and for humans, typically the height of these trees will be something like millions of years. And as you've probably seen in Wilder's talk and also Jan's talk, so these genetic relationships change as we move along the genome. So in one part of the genome, you can describe genetic relationships using a tree structure, but then if you move along the genome, then this tree shape will change due to recombination. And I've tried to sort of highlight this a little bit in this matrix over here. So this matrix shows individuals as rows and polymorphic sites as columns, where I'm assuming that polymorphic sites are biallelic, so I only have either the black allele or the white allele at any given position. And I've sorted these rows in such a way so that at the left-hand side here, the mutations are ordered by inclusion. So you can see if I have this tree here, and I drop mutations onto these branches, then these mutations will be sorted by inclusion. So if you can sort these patterns by inclusion, that is sort of indicative of that there's an underlying tree that relates these people. And then if you sort of move further to the right, so let me get a pointer. So if you move further to the right, then you'll see that mutations will start to sort of not respect this order by inclusion anymore. And so, for instance, if you look at the rightmost position here, then we clearly have a mutation here that isn't ordered by inclusion relative to the mutations on the left-hand side, and that sort of shows us that recombination has broken up our patterns here. Okay, so, and the key point I want to make in this slide is that the long list of evolutionary processes that we've listed in the previous slide shape genetic variation through the underlying genealogy. So really, these processes impact the genealogy, and the genealogies impact the genetic variation. Okay, so in statistical terms, I think we can think of these genealogies as sort of an unobserved latent variable that usually we want to integrate out. So what we will try to do is we'll invent informative statistics that summarize our data in a certain way, and then we'll try to integrate out all possible histories to get to infer anything about our past. And so if we could do this step really well, and sometimes we can, then this is sort of a really nice way of doing this. But unfortunately, often this integrating out part is quite difficult, and we'll have to simplify often and approximate to achieve this, which often means that we lose quite a lot of power in inference. Okay, so today I want to talk sort of about whether we can go the other way. So I'd like to talk about whether we can go from our genetic variation data, reconstruct our genealogical relationships back in time, or perhaps a sample of these of versions of these genealogical relationships, and then take this as a given and infer things about our past given these genealogies. So in principle, I think this sounds like a good idea. And the real question is basically how well can we do the first step, and then the second step will be sort of if we can do the first step really well, the second step will be really powerful. But there are obvious challenges, and so I think that's what Jan mainly talked about in the second talk, is that it is challenging to do this first step for the sort of increasingly large data sets that we have, where for humans, I think we have data sets with more than tens of thousands of individuals and hundreds of millions of mutations genome-wide, and that's just a huge amount of data that we somehow have to process. So that it's quite a challenging thing to do. And but in principle, sort of inferring genealogies is an old problem, and there are lots of methods out there, but again, sort of only few can scale. And I've listed 4 methods here that actually do scale. So Argweaver, I think published in 2014, was sort of the first method, I think, that really showed that you can do this for more than a handful of samples. And then since then, there have been other methods, including tsinfer, which is the method that I think Jan talked mainly about last time, and relate, which is a method that I developed. And both methods were published in last year and scaled to large sample sizes. There are sort of small nuances between these methods, so that they all do the same thing in that they infer a genealogy, but there are sort of small differences, and perhaps I can highlight one, which is that Argweaver actually tries to sample ancestral recombination graphs. So that's the object down here, which is sort of this graph structure that relates present-day individuals back in time and has these coalescence events, which is when 2 lineages come together, and these recombination events, which is when one lineage sort of branches into 2 lineages, indicating that at this point you receive genetic material from sort of 2 different parts of the tree structure. So this is what Argweaver tries to do, and the other methods tend to infer something more similar to a sequence of trees, where each tree shows the genetic relationships in one part of the genome. And so these 2 representations are related. They're not quite equivalent, but they capture sort of a lot of the same information. And so when I say genealogy, I mean any of these representations, really. And I guess the other key difference is that these other methods typically start with inferring a point estimate of the genealogy, and then perhaps later we can be more ambitious and sample, whereas Argweaver actually samples ancestral recombination graphs. But on the other side, both tsinfer and relate are much more scalable than, for instance, Argweaver, and can scale to thousands or tens of thousands of samples.
Is there some advantage of actually having an ARG? I mean, I can understand sampling being useful and helpful, but it seems like the tree sequence is sort of strictly more informative because you have a per site information.
So Well, so you can encode an arc in a tree sequence. At least that's what I understand. But in practice, if you do inference, the sort of tree sequence that you get out is not quite equivalent to an arc. And the reason is that an arc explicitly sort of tells you where the recombination event happened, which is not so obvious if you have sort of a sequence of trees. So in that sense, an arc is more informative in some ways.
But if you sort of specify a sequence of trees and SPR moves between them,
yeah, then that's fine. Then it will be equivalent.
Okay, thanks.
Okay. Yeah, so there are these methods, and I have only one slide really on relate here. So this is a method that I helped develop, and you can go on this link to download it. And just to sort of list a few key features, I guess. So it's fast and accurate, which is sort of the necessary conditions for this method. But also it's robust to errors. So we found that this was actually quite important to us. So it will be robust to sort of sequencing errors as well as phasing errors and like misspecification of ancestral alleles, which is required by relate and these kinds of things. And as a third point, it infers branch length, and it actually infers branch length jointly with a demographic history. And this is a nice feature, I think, in the sense that often you'll find yourself wondering whether the sort of signals you see are caused by the process you're interested in, or whether sort of some strange demographic history could have led to the same signal. And in this way, you've got the demographic history sort of built into the trees, which makes these types of analysis somewhat easier, I think. And then the fourth point is that there are lots of add-on tools for various types of analyses. So that's really what I'm going to focus on today. And this is not really, so this is not just specific to relate. Other methods also have quite nice tools. And really, I think what is somewhat important to say, I think, is that to a large extent, these methods are interchangeable. So there are nuances and there are differences between the methods, but sort of at least the key ideas about what type of information is encoded in these trees and how we get to these types of information is sort of shared across these methods. Okay, so I think it might be nice to start looking at some real data and some real trees. So this is a tree I built using Relate for one position or one region in the genome, where I've taken European individuals and British individuals from the 1000 Genomes Project dataset, somewhat downsampled, and also one Neanderthal individual who's over here and is something like 40 to 50,000 years old. And the point I really want to make here is that already looking at just one tree is actually quite powerful and tells us quite a lot about the history of our sample. And genome-wide, we have sort of millions of these trees. So aggregated over all these trees, we really have a powerful way of looking at our history here. So if you look at this tree, so the gray line sort of shows you the shape of the tree, and the sort of timings here are determined by the posterior mean of the branch length, and these error bars show you the quantiles of the coalescence times, and the red dots here are mutations that that happened in this region of the genome. And I've included this dashed line here, which is sort of approximately at 100,000 years ago. And what you'll notice, hopefully, is that below this dashed line, you'll tend to see Yorubans coalescing with other Yorubans and British individuals coalescing with other British individuals, but sort of no or few coalescences across these groups. So for instance, here you have a clade of only Yorubans, and here you have a clade of only British individuals, and there are very few across these groups. But then sort of once you cross this dashed line, you'll start to see coalescence events happening across these groups. And that's sort of showing you that these groups probably split something like 100,000 years ago. And then once you cross this boundary, these lineages start to coalesce more randomly. And sort of this will disrespect the labels that you put at the bottom of the tree. And so that's the primary reason why we see sort of not like one block of British individuals and one block of European individuals or something like that. It's simply because these groups are relatively young compared to their evolutionary history. Okay, and so actually, so the interesting thing I think is that even if you only look at British or only at European individuals, so the time to the most recent common ancestor of the sample will be more than 1 million years old. So there are Europeans falling on this side of the tree and on this side of the tree. So the most recent common ancestor of all Europeans is 2 million years old in this part of the region. Okay, and then we can also look at the Neanderthal here. And I think this might be an example of integration into non-Africans, where so these are British individuals. And if you remove the Neanderthal, you'll get a really long branch here extending to 1 million years ago, which could be Neanderthal integration into non-Africans. Okay, so that's just one tree. This is another tree. And here I've plotted the tree for a mutation in the that's known to be associated with lactose tolerance in adults. So usually we'll have some sort of lactose tolerance as children, and we'll lose that tolerance over time. But for carriers of this mutation, it seems like those are more lactose tolerant as adults. And this is sort of a quite famous example now of strong positive natural selection. And you can sort of see that, so the red lineages carry this derived allele for this mutation. And you can see that this mutation sort of arose fairly recently and then outcompeted all these other lineages to a quite impressive present-day frequency in British individuals. And similar events have been seen in other parts of the world as well. Okay, and perhaps, so the third example is perhaps more of a cartoon. So we could think of mutations being of two types. We might have a blue type and a red type. And so one thing we can see is that these trees will date mutations. So if you put them on these branches, we'll know that they happen somewhere between sort of the upper bound and the lower bound. And so having these dates will mean we can infer mutation rates through time. And so with this example, we might find that there was an elevated mutation rate here because these red mutations cluster in this time period. So we could also look at this type of information. Okay, so now we've looked at sort of 3 examples. And so what's the plan for today? I think the plan should be that we will now try to sort of formalize this a bit. So far we've looked at trees and sort of described what we've seen, and it was all a bit sort of anecdotal. And now I think what I'd like to do is sort of think about how we can use statistics to sort of do a bit more formal inference of these types of processes. Okay, and so there will be 3 sort of broader topics. So first, I'll talk about the duality between statistics of genomes and genealogies. So this part will really be about trying to see how we can sort of map over the types of analyses that we've done without genealogies to sort of a world where we have genealogies. And then in the second part, I'll talk about how we can get at sort of population histories. And then finally, I'll talk about natural selection. And sort of another thing that's sort of quite nice about this type of approach, I think, is that all these inferences will be derived from the same version or same reconstruction of history. And that will make these inferences hopefully more self-consistent and easier to compare to each other, which is hopefully a nice feature to have. Okay, so let's start with the first topic. So this is work led by Peter Ralph. So I'm not involved in this work, but it's sort of really nice to illustrate the connection of sort of statistics we compute using a genotype matrix, for instance, and statistics we can compute using a genealogy. So to start, I think this is mainly sort of a recap of what Wilder's talk was about. So a tree sequence is a way of storing a genealogy efficiently. So it's sort of a data structure for storing genealogies. And so using this data structure is sort of a fancy way of storing a VCF file. It's a lossless and highly compressed way of organizing genetic variation data. And as such, any statistic you can compute using a VCF file, you can also compute using a tree sequence. And the benefit is that it's sort of a smart way of storing that type of information, and so it will be extremely fast to compute lots of these statistics. So this is shown in this plot down here, where on the y-axis is runtime for some statistic. I forgot which statistic exactly they computed here. And on the x-axis, I've got sample size. So these two methods here are based on computing from a genotype matrix. And this line here does the identical job, just using a tree sequence. And you can sort of see that. So if you go up to 1 million samples, then you have sort of 3 orders of magnitude improvement in speed, which is huge. So that's the benefit of doing this with tree sequences. And if you go on the website, there's sort of a statistics tab, and then if you click on it, you'll see that lots of these classical PopGen statistics are implemented. So you'll be able to calculate the mean nucleotide diversity or the F statistics for admixture and so on. Okay, so that's great, but really you want to sort of see whether we can actually get something out of the genealogies themselves. And so Peter has sort of shown that there's a natural correspondence between summaries of these genotypes, so statistics you compute on a genotype matrix, and the statistics that you compute using these trees, so summaries of tree shapes. And so this is the natural correspondence. So branch statistics is the name for statistics of these tree shapes, and site statistic is the name of our sort of more traditional statistics of genotypes. And he's sort of shown that in expectation under some model, you can always construct a branch statistic, such as statistic working on trees, that in expectation is identical to the statistics of genotypes. So to give an example, we could think of the statistic of genotypes as being the mean nucleotide diversity. So what is the mean nucleotide diversity? For 2 samples, this is just the number of genomic differences in this part of the genome. So in this case, it would be equal to 3. And so what would be the branch statistic equivalent? It would be the mean time to the most recent common ancestor. So the time to the most recent common ancestor is the height of this tree. And you can sort of think of this as if the number of mutations on such a branch is proportional to its length, then you'll have that these 2 quantities will be equal up to a constant. So there's this natural correspondence, and it's actually not that much of a surprise in the sense that lots of these statistics of genotypes were originally sort of proposed with sort of this relationship in mind. So they were meant to capture some property of the genotype, of the genealogies, but the problem was that we couldn't construct these genealogies, so we had to sort of resort on computing statistics of genotypes. Okay, and this is an example of this sort of in action. So here on the y-axis, I've got mean time to the most recent common ancestor, or mean nucleotide diversity. And so the black lines here correspond to the mean nucleotide diversity, and the red lines to its branch statistic equivalent, which is the mean time to the most recent common ancestor. And at these dashed lines here, there are these strongly positively selected sites. So at these sites, we would expect that genetic diversity would reduce because some haplotype has been swept. I think in this case, in most cases here, to fixation. And I think what this quite nicely illustrates is that the branch statistic in this case is much less noisy compared to the site statistic. And so in principle should be much more powerful, so that the signal to noise ratio should be much better using the branch statistic. And so this is using simulated data and true trees actually. And down here is a more sort of real world example from the Arcweaver paper. And so what they did is actually really nice. So they looked at genes in the human genome and looked at sort of the mean nucleotide diversity in the exons and introns. And what they found is, firstly, when they did this using the mean nucleotide diversity and TMRCA, to sort of see that the patterns are extremely similar. Again, sort of showing that there is this natural correspondence between these 2 quantities. And well, what they observed is that there's sort of reduced diversity in these exons. And they followed up some analysis and suggested that this is due to background selection. So linked selection, linked negative selection. Okay, so that's the first part. So I think now we have these tools to map over our previous statistics into sort of the genealogy world. And now I'd like to move on and talk a bit about how we can do genealogy-based inferences of population histories. And this will be mainly things that we've developed in the Relate paper, plus a little bit of a sort of small new method and some new data sets as well. Okay, so I already mentioned this a little bit, but so one thing that Relate does is it infers population histories or population size histories jointly, as well as the genealogies. And so that's what I'm showing here. So I've simulated this sawtooth history. So that this thin black line is the true history that I simulated under. And then so I can get the true trees out of this simulation. And then when I use my statistic to estimate the population size history, I sort of perfectly recover this this ground truth. So that's good. And now the question is, how well can we do with inferred trees? And the orange line here is inferred when I have a constant size population size prior. So here I'm trying to fit sort of a constant size population, and it's sort of trying to do that and realizing that actually it might not be constant, but it sort of has to be a bit more flat. And then if I jointly infer the population size history using Relate, I get sort of a much better fit, which is the red and the blue line. And actually, even if I only have 3 diploid samples, I'm doing exceptionally well, I think, here in this case. And I think I should note that the y-axis is not in log scale. And sort of to highlight a bit more the point that it matters, that demographic history matters, I have this example here, which is part of a new method that I'm working on, which is showing that demographic history is actually reflected in inferred mutation ages. So to recall, we can date mutations by placing them onto these trees, and then we'll know that this mutation happened somewhere between this point and this point. And actually, if we have a constant mutation rate and a neutral mutation, then all we know is that its age is uniformly distributed on this branch. So we can date these mutations, and I can show that mutation ages contain the demographic history. So here I've calculated firstly using Relate. So these are Relate curves. The population size history between a British individual and the rest of the British individuals in the 1000 Genomes Project, where on the left-hand side, I've used a constant size population size prior, and here I've jointly fitted the population size. So this is using our previous method. And then using this new method that doesn't use the trees, it only uses mutation ages, we can sort of recover these curves almost perfectly, showing that the mutation ages actually reflect, the demographic history is reflected in these mutation ages. Okay, and so in this method, we're sort of trying to exploit this fact and infer these population size changes. And the idea is that this will be quite a flexible approach that can be used on unphased data and also low coverage data where we will use read information directly. Okay, but this is still under development. And so I can apply Relate to human data sets to estimate these human coalescence rates. So here I've looked at the Simon's Genome Diversity Project, which has about 280 individuals from 130 populations. And I've a bit arbitrarily sort of grouped them into 7 groups here, and then calculated the within population size and the across population size. So the solid line here corresponds to the within group, and then the dashed lines correspond to the across groups, meaning that when the dashed lines sort of start to diverge, that's sort of indicative of these groups splitting. And how to read these plots is on the y-axis, you've got the population size, and on the x-axis you've got time. I should have said earlier. Okay, and basically what we see is what others have seen using other methods and other data sets as well, is that there are sort of these out of Africa bottlenecks in non-Africans, which is actually really deep in people from the Americas and Oceania, and sort of absent in Africans. And then, yeah, you can sort of infer these split times of these groups as well. So that's sort of, yeah, go ahead.
So we do have a question from Gregor. He asks, what are the minimal data requirements to infer any over time? Do these requirements vary between large and small actual NE? Also, your NE stop at 10 squared to 10 cubed in the past, but let's see. That's not quite true for these ones. I think he was looking at the previous slides.
Yeah, so here they stop at 1,000 years ago, which is like 30 generations ago or something. Right. Well, so generally, you need high coverage data to first infer these genealogies. And then, so the sample size requirements is, well, so I think in simulations, it seems to work already for a handful of samples, so 3 to 5. I think usually it's better to have more. And especially if you want accurate estimates of sort of more recent population size changes, then more samples will certainly help. So here I've got a few hundred, and I think it works well. Yeah.
So now that we've sort of interrupted your flow, all we've really heard so far is that the relate method is mapping mutations onto genealogies, and this allows us to infer things. Can you just say a little bit more about the method, or are you going to catch that later?
About the relate method?
Yeah.
Oh, I see. Yeah, I'm planning to go very deeply into the relate method, but I can, I guess, try briefly.
I mean, does it have an objective function, or is it also sort of described as an algorithm?
It is described as an algorithm. So the shared part with sort of the tsinfer approach is that we've also split it inference into first inferring tree topology, and then as a second step inferring branch length. And so that the tree topology step is more described as an algorithm, and has sort of heuristic steps to speed up this inference. And the ideas are quite similar to what tsinfer does. So we use a Lean-Stevens type hidden Markov model to copy from each other and work out relationships from there. Cool. And then the branch length method is more likelihood based. So we use a coalescent prior, a constant size mutation rate model, and sort of try and sample from the posterior distribution.
Okay. I mean, I guess this is a different talk, but I wish I could hear it, like the combination of a coalescent prior plus recombination sounds interesting. Yeah.
So in this case, we will infer branch length separately for each tree. So we'll sort of throw away the information about recombination. And we've tried at the start to incorporate recombination in this inference, and found quite quickly that it's quite challenging, mainly because you don't know exactly where the recombination event happened, and you'll undercount recombination events. And so that makes the whole thing a bit tricky. And I think other people have found the same problem.
Okay, I see. So you assume, I mean, in a sense, you're sort of like aggregate, like everything gets its own branch lengths, but then in the end, when you're calculating these sorts of NEs, that aggregates over all the different trees. And so it sort of probably smooths out some stochasticity that you would have in the individual branch length inferences.
Yeah, that's the idea.
Okay, cool. So Gregor continues and asks, can you estimate current today's NE? And I guess you sort of said, well, you would need a lot of data to do that.
Yes. So if you want very recent population sizes, I think you'll have to explicitly use LD structures. And so there are, I think, other methods that might be more suited to that than this.
Yeah, we also got a comment from Yile that says, I think current NE can be better estimated by IBD segments, such as Browning's method on IBD NE. Anyway,
Yeah.
I don't want to interrupt you too much, but it's very interesting.
Yeah. Okay. Well, thanks for the questions. So here I've sort of grouped these samples into these 7 groups, but I can also sort of not group them. And so what I'm showing here is a series of matrices where these are sort of individual by individual, and then the color shows you the effective population size. And so if you go back to 1 million years ago, by eye at least, we can't really see any structure. And then sort of going forwards in time, starting perhaps 100,000 years ago, we'll see some structure emerging. And so at the bottom left here, I've got African groups, and the rest are non-African. So we see that Africans and non-Africans start to split something like 100,000 years ago. And then going sort of further, closer to the present, we'll start to sort of see more and more structure. And then perhaps when we look at the matrix 5,000 years ago, and then compare it to the matrix that sort of calculates geographic distance between samples, you'll, I think, see quite a nice correlation between these 2 matrices, which is perhaps what we expect. And even in the most recent time bin, we sort of see quite a lot going on the diagonal, and then not very much going on the off diagonals, suggesting that there is actually quite fine scale structure encoded in these genealogies. Okay, and then I guess I also want to talk about a non-human example. So here we've got 50 wild mice sampled in India, Taiwan, and France. And just to sort of give a bit of background on this species, so it's thought that these wild mice evolved somewhere in Central Asia, and then sort of spread to different parts of the world. So we've got samples from India and Taiwan, which are both classified as Mus musculus castaneus. And then we've also got some mice from France who are classified as this other subspecies, Mus musculus domesticus. And if you look at our species tree, so we've got famulus, Caroli, and Spretis as sort of mice that act as outgroups to this sample. And then we can see that the relationship of these mice is that it seems like the Taiwanese and Indian mice are more closely related to each other compared to the French mice. And the sequencing here is about 10x. And yeah, and phasing is sort of done within sample, without reference panel, and so on. So it's quite different to our human examples. And so if we look at the demographic history, here on the left, I've got a plot that's sort of a Taiwanese perspective of the population, of the demographic history, where sort of this line here is the demographic history of the Taiwanese mice. And you can see that there's a deep recent bottleneck. And then these other curves are sort of the cross coalescence rates with Indian mice and French mice. And we can quite nicely see that we'll first merge with the Indian mice and then with the French mice. Okay, so that's sort of the broad scale picture. And then we can focus a bit more on the Taiwanese mice. And that's what I've done on the right-hand side here, where on the x-axis, I've plotted sampling distance between these mice. And then on the y-axis, the effective population size between pairs of samples. And what we can see is that mice sampled close to each other seem to have a relatively small effective population size. And this increases as we sort of move further away. So if you sample 2 mice a few hundred kilometers away, their effective population size tends to be magnitudes of order bigger than when you sample them close to each other, suggesting some sort of gradient in structure on Taiwan. And you can sort of track how this structure changes across time. And so you'll see that it sort of weakens and then sort of disappears something like 2,000 years ago completely, perhaps suggesting that there was sort of a major migration event or peopling event of these mice on Taiwan. Okay, and another example of what we can do is look at mutation rates through time. And this is another example using the mice of the previous slide. So what I've done here is basically use the idea that I showed at the start of the talk, where we look at sort of clusters of mutations at specific time points and use that to sort of infer changing mutation rates. And what we've done here is we've grouped mutations into 3 categories. So the first category is a CG mutation, C or G mutation, nucleotide mutating towards an A or T nucleotide. So we'll call that mutations that go from GC or away from GC. And then another group where we mutate towards GC, and then 2 groups here that correspond to neither. And when we do that, we see that relative to today, the mutation rate of towards GC increases towards the past, and the mutation rate from GC sort of decreases towards the past. And then in the neither category, we sort of stay constant. And we think that this is a sort of signature consistent with GC biased gene conversion. So to explain quickly what GC biased gene conversion is, it's this process that happens in the same sort of process that will initiate recombination. So in meiotic recombination, you'll have these double-strand breaks. So one of the chromosomes will sort of break, and then sometimes this will result in these crossover events, which we understand as recombination. But a lot of the times, it sort of results in these non-crossover events. And so when you end up here, you'll end up having to sort of repair this blue chromosome against this red chromosome. And so sometimes you'll have polymorphisms in the track that you've sort of broken up here, meaning that you'll have to choose either the G or the A at this position. And GC biased gene conversion simply says that there's a bias when you choose A or G in favor of G or C. Okay, and so why is this consistent with GC biased gene conversion? So this process favors mutations towards G or C. And so that means they will be relatively overrepresented the further back in time you go. And then the mutations that go away from G and C will be underrepresented because they will sort of be selected less often and so have a higher chance of sort of vanishing in the population. And if we divide the genome up into sort of high recombinant regions and low recombinant regions, we can see that this effect is many times larger in the high recombinant region, which is also consistent with the idea that this is GC biased gene conversion. So in this case, actually, what we ended up finding is not actually a change in mutation rate, but sort of a confounding caused by GC biased gene conversion. Okay.
So we have a question about this. Have you tried validating the timings of features in these mutation rate curves using simulations? In particular, he is interested in understanding how much the estimates are impacted by not knowing where on the branch a given mutation occurs.
So not knowing where it occurs, we sort of integrate that out. And we have done a few simulations, not specifically for GC biased gene conversion, but sort of for mutation rates changing in a subcategory of mutations. And we've shown that we can fairly well recover this kind of signal.
And I think because I was distracted by that, I missed your explanation for why this is gene conversion and not just a change in mutation rate, like a mutation signature, basically.
Well, this is basically consistent with GC biased gene conversion, right? So, I mean, it could be a change in mutation rate, but we think this is like combined evidence, especially that this is this effect is very strong in recombination hotspots, I think is quite strong.
Okay. That sounds like an important additional piece of it.
Yeah.
Okay, thanks.
Thank you. Okay, so that's what I had to say about population histories. And I think in the last part, I'd like to talk a little bit about positive selection. And so in the first part, I'll talk about monogenic selection, so selection on a single mutation, and then later on, hopefully extend that to selection on traits that are governed by multiple genes and multiple, so hundreds of genes, polygenic selection. Okay, so this first part will again be partly methods we had in the Relate paper, and then also a really nice method developed by Aaron Stern that allows you to actually infer selection coefficients using these genealogies. Okay, and perhaps we can sort of relax for a minute and again start looking at trees just to sort of get an idea of what we're looking at. So here I'm plotting the tree for the EDA gene. This is again a relatively sort of classical or well-replicated example of strong positive selection, where again, we see sort of that this mutation is relatively young and outcompetes lots of other lineages to our impressive present-day frequency, in this case in Chinese individuals. And there are other examples. So this is an example where mutation associates to light skin pigmentation has swept, I think, almost to fixation in this case, mainly in West Eurasia and also South Asia. And this is a non-synonymous mutation. And again, you can see that it seems to spread really rapidly in these groups. Okay, so we have these selection signatures, and now the question is, how can we actually quantify this? And what we sort of want to do is try to measure how quickly a mutation spreads. And for that, I think it's good to understand what happens in the neutral case. So that's what I'd like to do first. And so I've got this tree here, and what I like to look at is when I slice it, when there are two lineages left in the tree, can I sort of predict how many descendants each of these lineages will have? And it turns out I can write down an analytical distribution for this, which turns out to be just the uniform distribution. So perhaps this is better illustrated using an example. So here I've simulated 9 trees using 10 tips, and these numbers show how many descendants this lineage has and how many descendants this lineage has. And the theory will tell us that it's sort of equally likely to have 9 descendants as it is to have 5 descendants or 4 descendants. And so this really says that we expect these unbalanced tree shapes. And so not many trees will look like 50-50, and a lot of the trees will be also looking like 1 to 9. So that's sort of the kinds of tree shapes we find in the neutral case. And we can use this to sort of now ask, what's the sort of probability that a neutral mutation that arose here spread to this frequency? And we can use that to calculate p-values. Yeah. And so this is what we've done. And here we're just showing that under null simulations of no selection, our p-values are well calibrated. And the really nice thing about this method, I think, is that this analytical distribution is unaffected by demographic history. And so this is robust to demographic history. And we can sort of see that in this QQ plot, our p-values really do seem to be uniformly distributed. And we've done some power calculations showing that we are well powered, especially in finding weak selection here with a selection coefficient of 10 to the minus 3. Okay, and this is a very fast method, and so we just applied this to every mutation in the whole genome. And what we find is that basically we recover sort of the usual suspects of selection. And what is, I think, really striking here is that the sort of really strongly selected loci seem to be actually relatively rare in humans. And that sort of falls in line with the idea that lots of these traits are governed by many genes, and so selection on any individual mutation should be weak, because the effect of every gene is weak in these more complex polygenic traits. Okay, but so the question is, how can we now aggregate sort of weak selection for any trait, and sort of how does weak selection evidence vary by trait? So that's what I want to do eventually. Before I go there, I'll also quickly talk about clues, which is a method that Aaron developed last year for inferring selection coefficients. And so this is a full likelihood method. So a likelihood method calculates this likelihood function, which is basically a summary of your evidence for your parameter, in this case, the selection coefficient. And the idea is that we can write down this likelihood if we knew the true tree. And in this case, we only know the inferred trees. And so what we will have to do is sort of integrate out all possible histories. That's sort of what I said at the start. And we do this by sort of calculating the mean over all sampled trees. So that's what his method does. There's a small problem in that we'll have to sample these trees given selection coefficients, non-zero selection coefficients. And the problem is that relay trees and most other methods will assume neutrality for all mutations. And so what we'll have to do is sort of correct for that bias, and that's what importance sampling does. So his method is an importance sampler, and will find the maximum likelihood selection coefficient. And this plot basically shows the performance of this method, where on the x-axis you've got this true selection coefficient, and on the y-axis you've got the inferred selection coefficient, and you can quite nicely see that you can recover the selection coefficient. Okay, and the other thing you can do with clues is infer frequency trajectories through time. So here I've looked at lactase and another randomly chosen SNP in the genome that have similar present-day frequency, and you can see that the lactase mutation clearly spread really quickly, very recently, and that's sort of what we expect, because we think that this is strongly positively selected. Okay, so we've got these methods now to quantify selection on a single mutation, and now we can ask, how can we sort of aggregate that kind of information across multiple mutations to look at selection on polygenic traits? And so in our relate approach, what we do is we look at mutations that are associated with a trait, and then simply ask whether the selection p-values are smaller than compared to some matched SNPs that we chose in some way at random. So we ask whether the selection p-values associated with the trait are smaller than what we would expect. And here's sort of an example trait, hair color, where I've divided the SNPs into either causing darker, being associated with darker hair color or lighter hair color. And what we see is that in Europeans, we have quite strong evidence of selection towards lighter hair color, as well as a little bit in South Asia. And we can replicate the signal if we replace relate p-values using IHS scores, but the p-values are quite a lot larger, suggesting that we have quite a lot more power in the relate method. Okay, so this is nice, but there are a few sort of problems or difficulties with polygenic selection. And this is just a sort of very short list of things that we have to be careful about. Firstly, we'll have almost always unbalanced power for different ancestries. So GWASs are predominantly conducted in sort of white European ancestry individuals, meaning that they might not transfer very well to other ancestries. And then we have also problems that phenotypes are correlated. So if you see a signal for one phenotype, we don't really know whether it's selection for that phenotype or a correlated response. And so I'm going to address some of these points now. So the first thing, for the first point, you won't get around doing a GWAS in a more diverse cohort, and that's this example here. So we looked at type 2 diabetes, where a group led by Anuba Mahajan, Andrew Morris, and Mark McCarthy, and the Diamante Consortium sort of did a very large meta-analysis of these GWAS studies and had something like 50% non-European ancestry. And this is important because type 2 diabetes is a common disease. It's affecting, I think, something like 1 in 11 adults, and it varies by ethnicity. So it really makes sense to look globally at how this disease works. And what they found is they found 338 independent loci associated with type 2 diabetes risk. And when we looked at whether we find any selection evidence, we saw that there's nominal evidence for selection for increased type 2 diabetes in African groups, but not elsewhere. And if we look at the mutation age of these mutations, we see sort of that in this African group here, we see that there are some mutations that are very young for their frequency, suggesting that there is positive selection acting on these mutations. And now the question is sort of what's going on. And so it's always a bit sort of tricky to put, and dangerous to put stories behind these things. But so there's one sort of long-standing hypothesis for type 2 diabetes that there's sort of there's a thrifty gene that sort of makes type 2 diabetes advantageous by sort of making it more efficient to process foods for sort of fat gains and storage. And that could be beneficial in some scenarios, perhaps, where there are sort of frequent food shortages and things like that. And so what we hypothesize is what happens if we look at other traits now in the UK Biobank and so sort of only at SNPs that overlap between two traits. And what we found is that if we looked at only SNPs that sort of overlap between type 2 diabetes and weight increase, then sort of we could capture the signal, and then the complement, we sort of couldn't see any selection evidence anymore. So this could be evidence that selection is acting, especially on the SNPs that are associated with both of these traits. Okay, and so now moving on. So recently, Aaron sort of extended his clues method to work on polygenic traits. And so to remind us, so his previous method inferred selection coefficients for individual SNPs. And now what he wants to do is infer these selection gradients, which is a parameter shared by a trait. And what the selection gradient really is it's a derivative of fitness with respect to trait value. So this product here will give you the fitness advantage of this particular SNP. And again, sort of he has this full likelihood method, and what it does is it infers the selection gradient, and we can see that he can sort of quite nicely recover this selection gradient. And he applied this test to the UK Biobank and found sort of a few traits, which is quite nice. So sort of the usual suspects, I suppose, including hair color and a few other traits. Okay, but so this is my last point here. So what was really nice about Aaron's method is that it can disentangle selection on correlated traits. And so that was one of the points that I mentioned. It's sort of difficult when we look at polygenic selection, is that we never know if we are looking whether selection is actually acting on the trait that we're looking at. So here, this is a simulation where there's selection on trait 1, and trait 2 and 4 are neutral, but they're correlated to trait 1. So trait 2 has an 80% correlation, and then trait 2 a 50%, and so trait 3 has 50%, and trait 4 has 20% correlation to trait 1. And then if we do our marginal test, so that's the test that we've done on the previous slide, then we sort of detect selection for each of these traits. And we know that actually detecting selection for these traits 2 to 4 is actually false, because their signal is only driven by selection through trait 1. And what he can do is sort of do a simple extension of his method and do a joint test. And if you do the joint test, then you'll see that you only detect selection on trait 1 and no selection on trait 2 to 4. So that's a really nice method. And he applied it to a few pairs of traits. So the first example he has is hair color. And when he conditions on tanning ability, you'll find that, so on its own, hair color seems to be selected, but then conditional on tanning ability, this selection signal is basically gone, suggesting that perhaps the selection signals we pick up for hair color is a correlated response to selection on a different trait, perhaps more close to skin pigmentation. And I should say this is in British individuals. And then type 2 diabetes is another trait that popped up here, where on its own, it doesn't show evidence for selection. But then if we condition on this trait, which is measuring sort of glucose levels in your blood, then you start to find selection for type 2 diabetes. So it's sort of the opposite scenario, where you're now conditioning on a trait and you find selection. So this could indicate that some specific pathways of type 2 diabetes are selected in this case. Okay, so to summarize, so we've seen, I think, across the 3 talks that we can now build these genealogies for huge data sets, so 10,000 people or more, and these actually capture quite a rich set of information about our genetic past, including sort of integration and mixture events, mutation rate evolution, trait evolution, and so on. But there's still scope for quite a lot of other methods that sort of use these genealogies, including sort of trying to use ancient genomes. I know there's been progress made in this region already, but I think it's good to stress this, and but also sort of more deeper analysis of varying types of selection, recombination, evolution, and so on. So basically, we need creative approaches to use these trees to answer a wide variety of biological questions. Okay, yeah, that's it from me. Thank you very much, and these are all the people who I have to thank. Thank you.
Cool. thanks for the wonderful talk. Very interesting. So we have a bunch of questions. So one is from Jun. He asks, was relate selection test calibrated for selection on standing genetic variation?
Yeah, thanks, Jen. So it is possible to detect selection on standing variants. At the moment, it will test for selection from when the mutation arose, but you can change that. So actually, if you run the software, it will give you a number of p-values where you condition on different time points. And so if you have a rough idea, I suppose, of when selection might have started, you can look at these different time points and look at those p-values as well.
Another question from Jun. Because both relate and clues look for evidence of selection on a SNP, are they used to detect partial sweeps but not for complete sweeps?
So yes, so that's a good question. That's true for relate, so the relate statistic. For clues, I believe you can look at selection that's on fixed loci, SNPs. I'm not sure if it's entirely robust at the moment, but in principle, that should be possible.
I also wanted to acknowledge that there was sort of like an ongoing thread that had to do with my question about differences between args and tree sequences. And Jan, I figure I might have just sort of read it to record it on the video. So Jan points out that inferred tree sequences don't typically have recombination nodes. And Peter points out that it's not, you don't just need to know the SPRs, you actually also need to know when the coalescences happened, not just the splits. So that's actually another point that I hadn't thought about.
Yeah, I see.
Let's see. And then, and the SPRs need not be unique anyway. And then, let's see. So Jan points out that this is a difference between TS for infer and relate. TS for infer plus TS date doesn't estimate a set of trees for each point in the genome, but essentially an arg. So there is one only date estimate per node. I don't know if you have anything else to add about.
Yeah, no, I think that's correct.
Okay. So it was neat to see that these tests were robust to demographic history, but I mean, I imagine that there are some sort of things that they are more. I mean, there's always like weird model violations that things can.
Yeah, so we assume, for instance, no structure and admixture and that kind of thing can easily confound this. Aaron did a bit more careful simulations actually, and showed that his methods are quite robust to these types of confounders. But yeah, it's true that you always risk.
That's cool.
Yeah.
Matthew asks, could relate or TS date infer recombination times to get the arg without destroying the scalability advantage over argweaver? That sounds like a big, hard question.
It is a hard question. I mean, so I can only speak about relate. We thought about this a bit, and I don't think it's impossible, but it is quite challenging. It's challenging for multiple reasons, I think, because firstly, you don't quite know where the recombination event happened along the genome, and then you have to sort of figure out, you don't have the arc, so you don't have an explicit way of showing where the recombination is, so it's a bit tricky.
But do I understand Jan saying that is something that TS date is currently trying to do, basically?
Yeah, that could be the case.
Okay. Just stepping back, like obviously there's tons of things to do with these statistics and inference and lots of cool stories to find. But like in terms of those sorts of things, what do you feel like the next frontier is, as far as sort of trying to take this from, like trying to make things a little bit more formal maybe, or I don't know.
So you mean
to do a bit better of a job just on the sort of arg inference side, or maybe that's not a focus of yours right now.
Yeah, well, so the arg inference side, I think, so if we simulate perfect data, then what we've seen, and I think Jerome and Jan and Wilder have also seen, is that the sort of 4 methods I've shown and other methods as well perform really well and quite similarly. So I think at that level, we've sort of maxed out a bit and sort of approached the theoretical optimum. But the differences are when you sort of deviate from that. So if you include errors, if you have ancient genomes and that sort of thing. So I think that's sort of the space where you can make progress. And then the other thing is these methods at the first instance infer a point estimate. So sort of getting confidence intervals on things and sort of measuring errors in our topology and things like that is needs to be more formalized, I think. And that's also another thing that we should be focusing on, I think.
Yeah, it's sort of that. I mean, that sounds like a tall order when like I feel like Bayesian analysis is out here and you all are like on the side of like scalability. It's going to be interesting. I mean, yeah. Okay. I think there's one more sort of thread that I'd like to pick up before we close, and that just has to do with the licensing of Relate. I noticed that it's not open source. And I'm just curious, like, what are the obstructions to Relate becoming open source?
Well, I mean, so part of the code is actually open source. So you can, there's a GitHub repository somewhere where part of it is open source. It seems to be sort of a policy that we've adapted in our department. And it's just, I think, a response to it is quite time consuming and difficult to sort of maintain these things. So that's mainly why we think. But I mean, it's freely available if you use it for academic use. And I've also added quite a lot of people to the GitHub repository. So it's not like we're protecting the code or like shutting the world off from the code or anything like that.
Okay. Yeah, I mean, it seems like the modern trend is more towards sort of getting more people involved in general. But anyway, that's interesting. Thank you very much for the wonderful talk.
No, thank you for having me.
And that's the end of this series of talks. We're going to take a little break for the summer and see folks back in a few months. All right.
Okay, thank you. Bye.